Good morning. Good morning, everybody. Okay, so we'll continue our coffee break with TRANSFAC. Grab your coffee. So, like I did, although it's again tea last time. So you can also take some tea and prepare your questions. And basically, I will do the following. You prepare your questions and put it, please, in Q&A here, and I will see when the questions will arrive, so I will try to answer them. I will start with some previous questions which I received during my previous talks and in between, so you can also send questions to our customer support or generally to me by email. And one of the repeating questions was how basically the estimation of affinity is done in our system. And let me start again from the very beginning. A little bit today we will have a little bit of a theory, but we have to do it and in order to understand how the things are going on in our system, how we predict transcription factor binding sites, and what is the technology behind it. So let me share my screen. Okay, over there, and I will start from my presentation. Just a moment. Okay. Here is, by the way, our website, but let me first start a short presentation for you. So what you see here, the transcription is a transcription factor, AP1. And as you know, TRANSFAC is a database about transcription factors and their binding sites. And now we're dealing with these very important proteins, which are living and functioning in the nucleus of the cell. They are binding to the DNA in a specific manner, so in special places which the transcription factors can recognize. We call them binding sites. And due that binding, these transcription factors can actually kind of anchor at the place, and then by the activation domains or other domains of those factors, they can interact with other factors, they can interact with transcription machinery, and they can regulate expression of the genes. And the TRANSFAC, a big part of it is actually concerned with recognition of binding sites in any sequence. And why do we need to do the prediction, the recognition of binding sites? Because so far we still very far away from knowing all functional binding sites in genome. And I talked about that before. So we have so many transcription factors in various cells which are functioning in various conditions, and they can bind to their consensus sequences in DNA. We have a number of technologies, experimental technologies, to understand where they are binding, to identify those binding sites experimentally. And now we have, high throughput technologies like ChIP-seq or TAC-seq, which allows us to really find many binding sites for a factor, in the whole genome. But still, our simple estimates telling us that still we are very far from knowing every possible sites in genome. Maybe we know just less than 1%, or even maybe 0.1% of all binding sites in genome. So we still need to predict. And how we do that in TRANSFAC, as well as in other databases, which you can see here, we basically collect so-called positional weight matrices, and you see them. One example of that matrix here in the screen. It's a positional weight matrix for a factor which is called AP1, and that was the picture I presented in the first slide. And this positional weight matrix is built from experimentally known binding sites, which we collect in TRANSFAC, or we grab them from experiments, high throughput experiments, like many other do in other databases. Then we basically count how many times a particular nucleotide was found in a particular position on that site. And then you see those cases when almost in all cases T is staying here in the first place. Then this letters is big in that logger plot. And if the position is kind of redundant, so it can be several nucleotides staying here, then those letters are small. So they not only present the motif, which this factor recognizing in DNA, but also presenting kind of a conservativity of a particular positions in that motif. And as you see that TRANSFAC is actually, we're making our efforts in TRANSFAC to collect as many as possible those positional weight matrices by integrating knowledge from many existing databases, from literature, which we are scanning, and by building these matrices by ourselves. So, and the whole idea of those matrices is actually to find new binding sites in genome. As I said, we still have to find them. So, because we don't know where those factors can bind, and if we don't know, we cannot actually explain how factors regulate the genes, regulate the gene expression. And but still, let me go a bit, in details how this all prediction works, because that's still a more often very frequent question to understand how we predict those sites, and whether our predictions are, first of all, whether they are correct, and how they correspond to real affinity of those factors to their targets. Okay, so let's go step by step. So first, as I said, we have a collection of known binding sites, which I aligned, like here. You have, for instance, 5 sites, which are experimentally known for a given factor. And we align them to each other according to the most conserved places in that site, using various algorithms. And then we count these positions. And from that counts, we can build a matrix which is called a count matrix. Basically, we have number of cases observed here, and then the first transformation we do, we basically normalize these counts by the number of sequences which were observed in that position, and then you have here the probability matrix and position specific frequency matrix, PFM, you can call it. So it's a frequency of a particular nucleotide in particular position. But then a certain transformation is useful, so-called the log score. So what happens here, in fact, as you may understand, that these probabilities, which we have here, are difficult to tackle, sometimes algorithmically, because if you want to find a new sequence which fits to that matrix, you basically have a sequence, and then you go through that matrix and you have to multiply these probabilities in order to get the probability of that sequence to appear. Why do we need to know probability? Because if we see that the observed sequence have a very high probability to be the binding sequence for that factor, we can claim that the sequence we found is a true binding site. But again, by multiplying those numbers, and especially when you have a long motif, or you have such places where you didn't observe anything, so zeros, they can really distort your algorithms. And that's why the useful thing is here to make a logarithm of those values and to use so-called pseudo counts. So as you can see here we don't have any irrelevant values which could happen if you have a logarithm of 0, because we add a small pseudo counts here, which kind of compensate our little knowledge about binding sites. So basically, we have our set of sequences, set of sites, and it's a limited set. It's not unlimited. So, for instance, only 5 sites, and our observation is, number of observations is very small. So we have to compensate the small number of observation by a certain pseudo counts. So that's why we got this logarithmic values, and then these values can be used further to estimate the score of a given sequence, and then by simply summing up those values when you see a sequence. So you take this matrix, you put it on the sequence, and then you simply sum up those values to each other, and you got a certain value. We call it a score, a log score. And if this value is high, then basically we can say that this sequence is very similar to the binding sites of transcription factor, of a given transcription factor. And another useful thing for the logots is actually, if they are negative, it means these values are very small, and they contribute, in a negative side. And basically, if you have a sort of score near 0, you may say that this site is nearly random. So okay, so that's how we calculate these logout scores and predict binding sites. By the way, in our actually more advanced algorithm, which we call match, we use additional weighting, which is weighting those positions in addition by a certain information content. So it's like a double weighting, telling that a certain position is really important, it has a high information, and we give a additional weight to those positions. But that's a special case, and I talked about that in my previous talks. But here, more traditional and way, which is also applied in other algorithms, which is called log odds, when you simply sum up different sum up position weights of consequent positions. And then we made some efforts several years ago, and it was published in this paper by Stegmaier and et al. In PLOS 1, where we basically generated a lot of random sequences and estimated probabilities so that a given score can be observed by a random chance. So we basically we calculated a p-value, so this famous p-value. So it's not enough to see just the score, but you have to say how probable to observe that score by random chance. And why? Because if it's observed and it's very rare, so p-value is 1 in -10, for instance, you can say it's a rare event, and that's why this must be a real binding site for a transcription factor. So we computed this score for many random sequences, and then we even made our estimates based on actually on the curves. Like here we used a mixture, a probability model to estimate those p-values, which allows us actually to do these estimations not by random generating random sequences each time, but by just estimating a certain parameters of those distributions, and then estimating those probabilities. So why again? So we want to know if we observe this score, how probable to have this score for that particular matrix by random chance. So, and now actually what I want to talk today, and I will show this in online in our system, that we not only compute this probability for a given position of a transcription factor to bind to that position, so basically probability of that binding sites to be a true binding site, but we can try to estimate a sort of affinity of that transcription factor to the whole region. Why should we do that? Because we never know how the true regulation of that particular gene is happening. So we know that if this sequence is, for instance, a promoter of a given gene, which is regulated by AP1 transcription factor, then in that promoter, we can observe several binding sites for that one transcription factor. And some of those binding sites are very strong, so which means they have very high log score. So and actually very low p-value, which means they are very improbable by random chance. So they are very good candidates for binding for those transcription factors. But there are a number of other positions in that promoter which have low scores, so lower than this maximum, but they still can be targeted by this AP1 factor. Why? And I was actually discussing that last time, because in many cases, transcription factors, they interact with their target sequences in the complexes with other factors. And for instance, there are many cases when these AP1 binding sites are occupied by AP1 factors, because there are other transcription factors recruit this AP1 to that place. So it's not really a very high affin to that particular place, but it's compensated by, its binding to that place is compensated by the synergy with other factors. But all in all, this promoter is regulated by AP1. It means that many sites in that promoter can contribute to the whole affinity of that promoter to that particular transcription factor. It could be either by one place, which is a very high scoring, or it can be a multiple low scoring sites which are distributed over the whole promoter. But in total, they contribute the same amount of affinity to the target, to that transcription factor, to bind to that promoter, and consequently to regulate that gene. So that's why what we came up with here and published in that paper is a so-called affinity score, which is given to a whole promoter. So not just to one binding site, but to the whole promoter or whole regulatory region, which we want to analyze for a given transcription factor. I'm sorry. So, and this is a formula how we calculate it. Basically, I'm not going into many details of that formula. It's very simple. So we have in each position of that promoter, for a given matrix, we can calculate the log score, and that log score is actually exponentiated first, and then it's summed up through the whole length of that promoter. So it's actually basically more or less the the sum of the weights over the whole promoter divided by the length of that promoter. Well, minus the length of the window of that binding site itself. So it's a kind of average affinity to that promoter for that transcription factor. And the good thing here is that this score is in fact not depending on the cutoffs of individual cutoffs of that particular positional weight matrix. So because if we would do the standard motif search, we would first run this log score through the whole promoter, and then we have to cut this score by a certain cutoff. And then we kind of have to choose which cutoff to use, and that's always a question. We don't know which what affinity, with real affinity, which factor is binding. And then if we select a high cutoff, for instance, somewhere here, we will find just one site, this one. If you select a low cutoff, maybe we find more sites, but then we find a lot of sites. So you see a lot of variability of your findings depending on which cutoff you choose. In the case of affinity score, we don't put any cutoffs on the position weight matrix. We calculate this log odds through the whole promoter. Then we sum up the whole weight of those scores, and we have one number, one figure for the whole promoter. And of course, we can apply this a new cutoff for that figure, a new probability for that figure, and that's something what we did, in our computations. What we had a question basically: if you have a random sequence, not a real promoter, but just a random sequence, and you apply this AP1 matrix to a random sequence, of course, you will find some matches to that motif, and maybe in some cases quite high matches, high scoring matches, and you would get this affinity score for random sequence. But if you have a real promoter which is regulated by AP1, we expect that this affinity score is higher than the average random sequence, right? So that's why we have to estimate how these scores are distributed randomly, and also make another efforts to estimate probabilities and p-values for those affinity scores. And what we did, and that's one example on one matrix, which is MyID. And actually, here is this affinity score, which is can be positive or negative, because it's average affinity basically over the whole promoter, and it has this multi-modal distribution, which is very interesting, because actually, which tells us that, and that's very clear why, actually, because in many cases, these matrices have very few, very conserved sites, very conserved nucleotides. So the matrix can be of the length of, say, 10, 12, 15, but maybe only 3, 4, or 5 nucleotides are really conserved. Like in these cases, we can say these two T and this A is most conserved ones. And maybe this one as well, the A. So which means if this particular site will be missing this particular very conserved nucleotide, you will get a very big jump in that score. So which means these high scoring sites have all 3 conserved nucleotides are really preserved in that site. This one missing 1 of them, this one missing 2 of them, and this one's maybe missing all of them. That's why their scores also so low. So, and then when we basically sum up these distributions, we can also make a parametric estimation of the probabilities, and through that we can estimate p-values of that affinity scores. So that's a little bit of theory today, just to tell you that that's what we are calculating. Those affinity scores, they're not just randomly chosen, but we made a very important and basic computations to understand how they work. And there are a number of other matrices I show here how affinity scores are computed. Okay, so now let me show you how this affinity score is actually used in real life in our tools. Yeah, so by the way, you can put your questions in Q&A, and I will, if I see the questions, I will interrupt my talk, or will finish it, and will actually try to focus my answers on your questions. Okay, I see first question, but let me finish first this part, and then I will come back to the questions. Okay. So what we will do now, we will basically look at our website. This is geneXplain website, and as you can see here, we can access directly all our products. So if you click on login, you see actually access to all our products which we offer. And let's start from this geneXplain portal, which gives you access to our databases. And then from databases, we will navigate to the tool for predicting binding sites in a given sequence. Actually, again, what I'm going to talk today, you have one promoter over your beloved gene, and then you want to find binding sites for transcription factors, and you want to predict which most important transcription factors can regulate your gene. And for that, we apply this affinity score, and I will explain how we do that. So let's first sign in our databases, and with this signing, it gives you a choice of the databases which you are subscribed to. So I'm now subscribed just to TRANSFAC and to PWM. And let's go to that database and search for a gene of my interest. So you see here this BRCA1, it's a very important gene, of course. Let's have a look at it. So you see in the database, we have information about BRCA1 and many different species. So for us, today is most interesting is human BRCA1. You know, this gene is a very important cancer. So mutations in that gene is leading to a high probability of having cancer. So it's indeed very important gene. And we have in our database information about that gene. For instance, you see here that this gene is associated with as a biomarker with many different tumors. And not only, actually, there are 35 different diseases where this gene is actually acting as a biomarker or published as a biomarker. And we have other information about, we have actually linked to human gene mutation database where you can get information about the known mutations in that gene. And here we have statistics about that mutation. And what is interesting today is to know, I'm sorry, where this gene is expressed. And this information is taken from actually Protein Atlas, and actually we also made our computations on those data, and it tells you expression value in different tissues. And as you can see, for instance, in testis, this gene has a maximum expression value. So that's why I'm interested here. Again, you know, coming back to my previous our previous discussion, we have paid a bit of attention to the simple question that the same promoter of the same gene can be occupied by different transcription factors depending on the tissue where this gene is expressed. For instance, in testis, this gene can be regulated by one particular set of factors, in another tissue by other factors. And that's why when you ask a question, which factors are regulating my genes, you have to actually tell in which tissue or in which conditions. And that's not, tissue is not the only condition which influencing the factors which are binding to the promoter. Maybe it's under certain stress or under certain, in certain cell cycle phase. So the condition is important to understand which factors are actually regulating my gene. That's why when we would like to search for binding sites in my gene and respectively factors which are regulating my gene, I have to take into account which tissue it is. So, okay. And there are other actually information here in our database about this gene. For instance, here it gives information about experimentally known binding sites in the promoter of that gene. So if you can see that, for instance, in that region near start of transcription, which is 0, there are a number of known binding sites for various transcription factors. Known means they experimentally proven by various experiments. So this gene is indeed very well studied. But again, as I said, known binding sites are still very few. So we still have to predict new binding sites, especially maybe in a tissue which is not yet studied in that particular experiments which were done to analyze binding sites for various factors. Okay, so you can if you have access to this database, you can look for more information about this gene. But today we go back to our search, and we want to select this gene, and we want to go to the match suit single gene analysis. So we want to analyze binding sites and predict binding sites in promoter of that gene. So now it will go to that new website, which is taking now your gene of your choice and preparing the promoters and enhancers of that gene for the analysis. So why I said enhancers? So important to understand that gene expression is regulated by many different regions in genome. The most important, of course, the promoter of that gene, but even the promoter can be different in different tissues. So here we have a selection for studied tissues. And if nothing selected means we don't know which tissue, we are actually agnostic to the tissue, more or less. And in that case, what system will do, it takes all known promoters for that gene, which are acting in different tissues, and also add potential enhancers, or not only potential, but known enhancers in those different tissues. So it will analyze not only promoter, but also enhancer regions which are associated with that gene in different tissues. If we select a tissue, like we want to select a testis, yes, then it will choose the promoter of that gene acting in testis, and also, if possible, if there are known enhancers in testis for that gene, then it will also select those enhancers. And then here we can specify which region we want to analyze, which length of the promoter, and in case yes, and by default we have this -500 plus 100, and but we can change it to up to -5,000 and plus 1,000 around start of transcription. So it's all about around the TSS region, so in the 5' region of a gene. So we can take the best supported promoter, or maybe most 5'. What does it mean? Actually, we don't. So there are experimental facts telling us that in a particular tissue, or maybe if we didn't choose a tissue, in all tissues, the same gene can be regulated by many different promoters, and some promoters are found, some start of transcriptions, they found actually very much in 3' region of a gene. They may even skip a certain first exons, and they are still observed in particular situations. They are really acting as start of transcription of that gene. And but most often, very often, there is one particular TSS which is best supported, which means, what means best supported? It's supported by EST sequences, by experimentally sequenced RNAs from that gene, and the majority of them are starting from that particular place. And then we can choose it as a best supported promoter. Or we can choose, for instance, most 5 prime, which means we are interested in that promoter, which is most left, most upstream. That make sometimes a lot of sense. But we choose best supported, and then we click next, and now the analysis is ready to start, and we'll launch the analysis. So it's what it will do now, it will first prepare these promoters and enhancers, and then it will take all 10,000 position weight matrices from TRANSFAC. Of course, it will select those ones out of 10,000 which deal with transcription factors which are for vertebrate genomes, so which is around 7,000. And it will skip those matrices which are, over low quality, we call it. So very small ones, or those which are not well described. So in the total, we have, in this analysis, we are dealing with about 5,500 various positional weight matrices, which are describing many transcription factors. And then, we start searching for those binding sites in those matrices and computing this affinity score. So we will not wait until it finishes. Let's have a look at previous result on this BRCA1. For instance, I have here, let me see, this one. Let's have a look at view results. So that analysis was done for testes, I guess. Wait until it will open. Okay, I will have a look in the meantime to my questions. Yeah, there is one question concerning what is the difference between the 5 prime and 3 prime most promoters. So I answered that. Again, just to repeat again, so one gene can be regulated by many different promoters, and this information we take from various databases, including actually ensemble database and including the Phantom database from Japan. So, and which tells us positions in genome where this given gene can be used as a promoter of the start of transcription. And then we look at for the most best supported, which is most often used in many different tissues, and we can also choose the most 5 prime, which is important because the most 5 prime actually that promoter is driving expression of the longest RNA from that gene, and it's the most complete RNA. And in some cases, it makes sense to take it as a to analyze because it's important to know the complete gene, complete RNA from which promoter is starting. The 3 prime, the most 3 prime is usually that promoter is used for a very strange variants of the genes which are skipping some exons, and that's also sometimes interesting to analyze. But in between, we got these results opened. And what we see here, 3 windows. So here at the left, it gives us the list of transcription factors. It found actually 17 transcription factors, which whose sites were predicted in that promoter of that BRCA1 gene, and which have a high affinity value with a good p-value, and the one which I explained in the beginning. And then, well, everything was started from particular matrices. So as I said, we run actually 5,000 different matrices, PWMs, and it chose these 5 PWMs which have a high affinity score to that promoter, and whose actually transcription factors are expressed in that tissue which we choose. And to these 5 matrices, 5 motifs, there are 17 transcription factors which are linked to that. Why is so different? So we have just 5 motifs and so many factors. Because, for instance, as you can see, this motif which is called GATA4 Q5_01. This motif was built on the basis of sites for GATA4. But in fact, this motif is very similar for all GATAs. And we make in our database extension, we call it a paralogous extension, telling us that if this motif was found in that particular region, we cannot say exactly which GATA factor is actually binding to that region. It may be GATA4, but it may be also GATA5 or GATA1, which is actually associated to the same motif. So all GATAs are very similar binding sites for GATAs. Although the factors themselves, they play different roles in different tissues. And particularly this GATA4, as you can see in that testis, this GATA4 is really highly expressed. All right, so and then if we go down here, and we see this window which shows the binding sites for those 5 motifs found in that promoter, and actually, and that's interesting case, because what you see here is that this promoter, or testis specific promoter, in fact located in the 2nd exon. So if you look a bit right to that gene, so we can zoom out a bit, and so you see here that the gene has exons and introns, and let me move a bit. And it's a very long intron, and then it has a first exon, but the testis-specific promoter is located in the end of this first intron, and actually in the front of the second exon. And that's something which you may actually miss when you just choose standard promoters, right? So we will focus now on that region. I will zoom it in a bit to see those sites better. What else you can actually do here? You can place some available information. For instance, you can put experimentally known binding sites. And as you can see, some of those binding sites actually coincide with those which we predicted here, although these known binding sites are known in various different tissues, but predicted are really focused on the tissue of our interest. And that's why sometimes you find a very good site like SOX9 here, but there is no experimentally known binding sites for SOX in that region, because maybe there were never done an experiment in that particular tissue for that particular region. Okay, so there are a number of questions. I want to come to those questions, but let me finish with that saying that actually we can have a look also at the report of that program, which shows you all the results in this reported form. It also shows this the whole algorithm, and it gives explanation how the affinity score is used here to select the best PWMs, and select the best motifs. And the important knowledge here is that we started from 5,000, more than 5,000 position weight matrices. We end up with 5 motifs only, which the algorithm found the most important for that region. So it did a lot of filtering, and the first filter was done by affinity score. So it goes through all 5,000 matrices, and it applied this matrix affinity score for each of that matrix for that particular one promoter. And it appeared only these 5 motifs have a affinity score with a probability with a p-value higher than a given threshold. So with that, we focus on those matrices, on those motifs which have a high affinity to the whole promoter. So it's a very special approach. And if you have your beloved promoters and you want to find all possible motifs, of course, you can apply all these 5,000 matrices, and then you will get a lot of matches, and you will not miss your motif of your interest. But if you really want to, have a first glance on those most important matrices and motifs which are dominating there and which are related to a particular tissue, then much suite is here for you to analyze. Okay. Let me then go to the to answer other questions. So we have a question. I have two genes which are okay. Probably they overlap. Yeah. So so I have two genes which are overlapping and located in the same region of promoter, but had different TFBS. I have two genes. They overlap in the same region of a promoter, but had different TFBS. Okay, as far as I understand, the situation is that there are two genes which overlap, or promoters of those two genes are overlapping. That happens. For instance, it could happen that there are two genes, and actually here we had this case in BRCA. There is another gene, NRB2, which goes in a different direction. So you see this BRCA1 goes from left to right, and this NBRB2R2, or it's even longer, it's actually long non-coding RNA gene, goes in a different direction. And in between, there might be a region which regulate both genes. So it's a kind of bidirectional promoter, some people call it. And it may have different binding sites. So some transcription factors binding to that promoter, they actually regulate BRCA1, but other factors are regulating that long non-coding RNA. And if you put this long non-coding RNA as a promoter here for analysis, it may happen that the program will find completely different binding sites here, because it will make a focus on a slightly different region, which regulating that gene, and it may happen that it will find other factors with a high affinity to that region. Okay. Another question, why it shows that heart tissue as well? The heart tissue as well. Where do you see the heart tissue? No, in that output, we have selected testis as a focus tissue, and we have two columns here. One column is actually for just tells you expression value in that tissue for that gene, and it has a color, like 25 is a maximum and 3 is a bit lower. So just to show you, this average. And then this column is very interesting. You can read about it in the report. It's expression difference. What it does is actually taking this expression value of that gene in all other tissues and compares this expression in testis to the average expression of that gene in other tissues. And if it's like 13.4, so it goes to the right, it means in testis it has a higher expression than in average than in all other tissues. If it's like minus 0.10, it's around average expression. In testis, it's this gene, LEF1, is expressed in average value, like in all other tissues. And if it's minus 5.0, it means this gene is actually in the testis is a bit lower expressed than in all other tissues. And the color here tells you whether this gene is tissue specific in principle. So red means that GATA4 is very highly tissue specific, and if you click on it, you see that actually it's not testis where this gene is mostly expressed. So you see the testis for this GATA4 is here. It's quite specific in testis, but not as specific as an ovary. So this gene is generally, in a general sense, is very specific, tissue specific. That's why it has a red color. We have it. And the yellow correspondingly less specific, and so forth. Okay, so that's why. And where did you have other tissues? Basically, there is a possibility in a different view to put actually more tissues here to have a sort of comparison of expression, but not in this interface. Okay, so there is another question. Thank you for a nice presentation. If I design a minimal promoter, what would be the basis for the screening and selecting the best TFBS to include in my minimal promoter? If I use match suite tool, in case if I use a match suite tool. In brief, how to prioritize those TFs to select the most relevant of them? Thank you. Very good question. So what I can tell you, so we had one recording, I think we can send you, if you send us email, where I was describing very specifically the question how to design the specific promoters. It's a part of another video. Generally, in we have another tool which is called Composite Module Analyst, which is a part of our genome enhancer actually tool. But actually, it has part of it is also presented here in Match Suite, but not in this Match Suite, which is for single sequence analysis, but the Match Suite for the gene set analysis. And the way how I would recommend you to do is actually, if you want to create a new promoter, an artificial promoter or enhancer region, which you want with a specific, properties. For instance, you want this promoter to be testis specific, right? So then what you would do is first you should collect a set of testis specific genes, and that you can do it here in Match Suite. So you go here and you simply select a tissue of your interest, let's say testis, and then it gives you a set of genes which are non-tissue specific. So basically these 552 genes are testis specific genes, irrespectively of whether they are transcription factors or just normal genes. They are presented here. Each dot here is one gene. So you can select those genes, and then what you do next, you apply, you select those genes. Let me do it like this. And then you proceed with those genes for the further analysis. And what it will do, and that's I was presenting some time ago, it will now apply this match, but not to any single promoter, but to all of the promoters of these 500 genes. And then it will apply a special tool inside, which is called Composite Module Analyst. And this tool is trying to find the best combination of binding sites, which are presented in the most of those testis-specific promoters. So the output of that program is actually a combination of matrices and their scores. And then what you want to do is, if you want to create your artificial promoter, you have to select those matrices, and this will be like 10, maybe 20 matrices for, let's say, 10 factors. and the build, artificial sequence which contains motifs, best motifs for those matrices, and the arrangement of those motifs can, again, this program, this this composite model can help you to find the best arrangement of those sites in your artificial promoter. So there are more details in that avenue of creating artificial promoters. If you want more details, just let me know. Just write me email, and it's a very, very interesting topic. Actually, I would be very happy to discuss it. Okay. Another question: could you explain what is the difference between p-values? Okay, so which p-values? I mean, so here we used several different p-values, and that's right. Maybe I have to explain a bit more. So first p-value here is a affinity p-value, and that's a p-value of that affinity score which I was telling in the very beginning. So we take a GATA, we take this matrix GATA 4 Q5, and we estimate affinity score for that matrix for the whole promoter, and that's affinity score here is 1.89. And the p-value, the affinity score is not shown here, but this is a logarithm of a p-value. So basically, it's a minus logarithm. So which means it's relatively okay-ish, I would say, p-value. It's not very low, but we have a cutoff of p-value of 0.5. So to be significant, and that's below the 0.5. So this is a p-value of a affinity score. Again, what is the p-value in this case? It tells you how probable, by random chance, to have such high affinity score of that matrix to the whole promoter. And if it's below this cutoff of 0.5, you can say that this affinity is significant, statistically significant, and you can choose this matrix and can claim that that promoter is a true target for GATA 4 or any GATAs which are estimated by this algorithm. We have another p-value here. I'm not sure if we have here in the we have again this affinity p-value, but if you go to view the whole table inside the platform, it has more columns. Again, we have affinity p-value, and we have a side score. We don't have here the side p-value, but you can find it more inside the platform. So this is a score for the given match. So as I said, it's not the affinity tells you the score for the whole promoter, and the side score tells you the score for the given match. So given position of that match. And then this score can be high, like 0.9 or 1, or relatively low. And we have also additionally a p-value, estimating a p-value of that score, of the match score. And then you can find it also. Okay. Another question. For one TF, there can be numerous PWMs, definitely defined by a number of the names, like AP1_0.2 and so on. Do these numbers correspond to a new version of PWM based on alignment of known TFBS? Question. Do you advise to use the last PWM built on the alignment of known binding sites as the most updated one, or we need to use all PWMs for TF to perform the prediction of particular TF? Very, very good question. Actually, that's a very important question, and a very frequent question, especially coming from the fact that Indeed, we have 10,000 matrices in TRANSFAC, or for vertebrates it's around 7,000, and which describe around 1,000 transcription factors. So in average, for one transcription factor, we have 7 and more matrices, and always there is a question, which matrix is the best? And the answer is we don't know. And this actually these abbreviations, like here, you see 0.2 or 11, or again 0.1, 0.7. This is just basically random number, not random, but we were curating the data from literature. And so when we found a paper describing SOX9 matrix, then it was 0.1. And then we find another paper and we give 0.2. Actually, right, so with the time, the number means most probably this is a paper which is a newer paper. But that's not necessarily the case. It can happen. It's just during our curation process, we found a later paper, maybe in the new time, and it may have a new number. So this number tells you nothing. Actually, more or less, it's just accession number. And what means this Q5? This means that matrix was built by ourselves, so by our team. And to build this matrix, we used binding sites, experimental binding sites with a quality 5. And the quality 5 means basically all sites. So, in the binding sites, experimental binding sites, they have different qualities, which we assigned based on experimental evidences. And you see here how many types of evidences might be to confirm a particular binding site. It can be just binding of a protein. It can be a functional assay confirming that this binding is really true, and functional assay confirming that this binding is actually leading to expression or regulation of that gene. So the more evidences you have, the higher the quality. And the best quality, it's a German system, is Q1, right? And the worst quality is Q6. So when we say it's matrix based on sites with quality 5, it means we took all known binding sites. If it's based on quality 3, then it will be Q3. And then, okay, to a certain extent, you can be more sure that Q3 matrices are really kind of better matrices, because they are based on sites with a higher experimental evidences, or with more experimental evidences. But saying that, I must say I would better just confirm and also support your last suggestion, telling that do you advise to use the last? Do you actually advise to use all PWMs? Yes, that's what we advise, and actually that's how the match suite is working. It takes all PWMs for one factor, all of them, and then it applies all of them to a given promoter. And maybe you have 15 PWMs for the same factor, for AP1, or maybe more, but only one PWM for that particular promoter gives you the best affinity. And then we choose one PWM from that experiment. Actually, the promoters which you're analyzing tells you the truth. So you never can be sure about experimental facts which are used to build the matrix. And the biological explanation of that is that each matrix represents a particular mode of action of that factor. And we don't know in which mode, maybe in a good mode or bad mode, this factor was binding to that particular promoter. And our approach, just apply all the matrices and let the algorithm select you that matrix which fits best to that particular promoter. This is a case for one single promoter, and for that we use the affinity score. And in case of analyzing sets of promoters, and that I was talking last time, there is another criteria, but again, we apply all matrices and then let the algorithm to choose the best matrix in comparing your set to the background set. That's another comparison. But again, the algorithm chooses the best matrix for you. Okay. Thank you for your question. Another question, Alexander, as I understand, the affinity score is a floating number. How does the program define the cutoff? Maybe I missed that. Every time, depending on the experiment, or it is set up on the average basis. Okay, maybe I was not clear in that. As I said, we estimate the affinity score, and for each matrix. But actually, selection of the matrix is based on affinity p-value. And so each affinity score for that matrix corresponds to a certain p-value. And we have a cutoff of p-value is 0.5. And all matrices which go below, a p-value of affinity score goes below that cutoff are selected here. And you can see it also on the motif table. Here where we have we have affinity score, logarithm of p-value of affinity score. So here you see both, right? So you have affinity score itself and the logarithm of the p-value of the affinity score. And then we have a cutoff of 0.5. So that's how we actually choose the cutoff by the p-value. Okay. Another question. It is important to have cell-specific analysis and not only tissue or organ-specific. Very good question. Thank you very much. Yes, that's something we are working on. And there is data in the Phantom database related to cell type-specific expression. And we have already data in our system, and actually it is present in the whole platform. Just to to let you know, what I showed to you is the interface, which is called Match Suite, which basically doesn't give you too much of the freedom to choose your parameters of the analysis. It does many things by itself, which is good. You don't bother with choosing the right parameters or choosing the right algorithm. But in fact, this tool, this Match Suite, is based on a platform, on geneXplain platform, and you can, if you have access to the platform and if you have actually account, then the platform is also accessible. So it will change, in fact, your view. You see that it's changing completely the view of the interface of that tool, and it gives you access to many different pipelines and also tools. And here among the tools and the databases, you can see, for instance, your question was about cell type specific analysis, and here we have this Phantom database. Yeah, you see, our analysis in Match Suite is based on Phantom tissue, which gives you the TSSs, the transcription start sites in different tissue types. But we also extracted from Phantom the cell-specific information, and it gives you information in which tissue and which cell type this particular promoter is acting. So, for instance, if I click on that promoter, it tells me this is well, it's a very general. Yeah, for instance, this promoter is acting in keratinocytes. This promoter is acting again in keratinocytes. So, so it it gives you a lot of promoters, this database, acting in different types of cell types. And we have a specific types of analysis among our methods. For instance, here is site analysis methods. So these are individual, methods which are used behind the scene of the match suite. And there is a method which allows you to analyze actually promoters of a particular cell types, and that's the whole set of methods. Maybe I will spend some time next time. It's a very good question actually to that, and I will explain how to use these individual methods and in a particular order to analyze different types of cells and tissue types. You can also generate your own types of conditions which can be taken into account by these methods. Okay, all right. So I think we have all questions answered, which I see on my screen. So yeah, thank you very much for your attention as well. So what do we have in chat? In the chat you see also some links from our technical support, where you can find the documentation to the match suite. Also, you can of course find us in the internet to send me emails, or you just send your question to the info@geneXplain.com, or just put your questions in a special form which we have on our website, where we put actually all our seminars and recordings of the seminars, so you can look at them. Come back to our website and you find many interesting information. Okay, so I already finished with my coffee, which was actually tea. And thank you for your questions, and see you next time. Thank you again.